Hi, and welcome back to Plantopia. Plantopia is the plant health podcast of the American Phytopathological Society, and I'm your host, Jim Bradeen. I'm a professor of plant pathology and associate vice president at Colorado State University. And today we're going to talk with Dr. Prasanna Kankanala. Prasanna earned her bachelor's in genetics with a minor in botany and chemistry from St. Anne's College for Women in India, a bachelor's in biology, and a PhD in genetics, both from Kansas State University. And as a PhD student, Prasanna worked under the direction of Professor Barbara Valent. Her PhD dissertation is actually titled Cell Biology and Gene Expression Profiling During the Early Biotrophic Invasion of the Rice Blast Fungus Magnaportha oryzae. So Prasanna is a plant pathologist through and through. And she's had a very diverse career path that has included industry, academia, and the nonprofit sector. And her roles have included senior research associate and scientist at Eden Space Systems Corporation, research associate at Kansas State University, and postdoc at the Noble Research Institute. And in 2020, Prasanna joined Trace Genomics. That's a company that's based in Ames, Iowa, that describes itself as a pioneer in the use of data science to activate the hidden insights in your soil. Prasanna has quickly moved through the ranks at Trace Genomics, and in February of this year was appointed Director of Research and Development. And in this role, Prasanna manages and drives research development of soil science and data science capabilities on behalf of the company, providing customers with biological and chemistry data interpretation. Prasanna has an impressive molecular biology and data science skill set, as well as an impressive publication record. She serves on the microbiome working group at the International Phytobiome Alliance. And today we're going to talk with Prasanna about her career path, her passions, and her experiences. And we're going to learn about the important ways that the plant and soil microbiomes impact plant health. We'll also discuss technological advances that are positioned to improve both environmental sustainability and the grower's bottom line. Prasanna, welcome to Plantopia.
Thank you, Jim. Thanks for the invitation.
Absolutely. We're delighted to have you here. And let me congratulate you on your recent promotion to Director of Research and Development. That's huge.
Thank you. Appreciate that acknowledgement.
So I'm curious off the bat, how did you get into the field of plant pathology?
That is a very interesting story. I had never imagined I would be in plant pathology. Growing up, I always wanted to be a medical doctor. I always was very enticed with the concept or the ability to heal people. So I tried my best to get into medical school back in India. I grew up in India, and I couldn't do that. It was extremely competitive there. And after that, I went into college. And when I went to college, I said, you know what, if I can't work with humans, I'll work with plants, because in my high school studies, when I took zoology, they made me cut open an earthworm and a cockroach and a frog. And I did not like cutting open animals. It was very, very difficult for me to do. So I said, I love plants. I'm going to go with plants. So I did a bachelor's in botany, genetics, and chemistry back in India. I did a 3-year bachelor program then. I was very fascinated with genetics. I think one of my genetics teacher and actually one of my friends in that course, they really inspired me. I really loved the way they spoke about genetics. So after finishing that, as I was trying to explore my career options, I always had that soft corner for human medicine. So when an opportunity came up, I actually came to United States to explore the option of a career in the medical field, physiotherapy option. I came in here for the physician assistant program is what I came to the United States for. I went to Jefferson College of Medicine in Virginia. It was a medical college associated with a group of hospitals there. I was very excited about that program. Before coming to this country, they gave me a structure for the program that I was very fascinated about. But when I came here, things didn't work out as expected. So long story short, I said, you know what, if I can't be with humans, I'll be with plants. So I ended up at Kansas State, and I wanted to explore a career in plant biology. And I wasn't And back in India, when we did plant biology, when we did botany, we did pathology, we did taxonomy, we did ecology, we did everything together. And I never realized that plant pathology existed as a career by itself. So when I came to Kansas State, at that time, I'm talking about 20 years ago, the university wanted me to do another bachelor's because in India, I did a 3-year bachelor program. They didn't know how to translate it into a 4-year bachelor program here. So they said, we want you still to do some coursework for a bachelor. So I ended up doing a second bachelor's in biology at Kansas State. And followed by that, as I was exploring options, I really liked the research projects that Dr. Barbara Wallen had in the Department of Plant Pathology at Kansas State. I met her in person and I really loved her so much. I said, you know what? If I really have to explore this option, I'd rather go with her. And I think what fascinated me the most was she worked on rice blast disease all her career, and I ate rice every single day. When she gave me 2 projects and she said, here are your 2 options if you want to come to my lab. And I read them up, the project and, the connectivity for me to be able to work on a crop that sustained me all my life was very exciting. So that's how I jumped into this field of plant pathology. I was still in an interdepartmental genetics program, but through the Department of Plant Pathology, and then rest was history. That's how I ended up in plant path.
It's amazing to me how many of our colleagues have a story that really starts with human medicine, and then through various fortunate opportunities, I'll say, these individuals have ended up in plant pathology. So that's a very interesting story, and we're happy to have you in the field. So after you graduated, you've had a number of professional experiences. You worked, I remember correctly, you worked first in industry, and then you worked in a nonprofit, the Noble Foundation, and you've also worked in academia at Kansas State, and now you're back in industry. So you've got really broad perspectives of what plant pathology is. And I guess my first question in this regard is, why are you in industry today? What are the advantages from your perspective of a career in industry?
My motivation for industry, Jim, has always been I wanted to see what I did translate into the field. I think that inspires me so much. I want to see the translational aspect, and that's what brought me to industry. When I graduated out of Kansas State, I was involved in a great research program there. I could have had a great career in research as an academic professional. I love teaching, but I've always asked the question, when will I see this information translate? How do I even think about translating this basic research so that, the user in the field, the farmer, can take advantage of it? And I felt like I didn't have that proper understanding and how that works. So I really wanted to go into industry and gain that experience. So at that time, when I graduated, there was this new startup that came into Manhattan, Kansas, where I was graduating out. And I went and interviewed with them. And I think that was a very good insight for me to think about, how do you think about research in an industry setup? Because industry is always oriented towards bringing a product out. And that experience there gave me the pathway to think through the lens of a customer, to think through the lens of commercial and sales and marketing, which does not happen when we are in the academic world. And the looking at things through the lens of the customer was the most exciting part for me when I was there. That was a biofuel industry. I was working on plant regulatory elements at that time. That was a short gig that I did, which was very exciting. It was a very good experience for me going from academia into industry. After that, I came back into a nonprofit setup, Noble Foundation. That is because that was, I really wanted to be in more in plant diseases and plant pathology kind of an area, because I was into biofuels and I realized that biofuels was not my piece of cake, truly. So I wanted to come back in there. And when that opportunity came up, I grabbed that opportunity. And in that nonprofit setting, it was really amazing because, there was no pressure that I had to generate these data sets to write grants for external funding. We had our own internal funding at Noble Foundation. So that gave me that space to explore. It gave me the space to explore new technologies, new methodologies, and to look at research from a very different lens. So I think that was a huge learning experience for me at the time. That is when I actually, for the first time ever, explored the world of microbiome. I was working on Sol-1 pathogens, which was it was Firminotrichopsis omnivora. It's called the cotton root rot pathogen, but on alfalfa, it's also called as Texas root rot. And there was a great reputation for that in that part of the country. And the reputation for that pathogen is it's a suicide for assistant professor jobs. So they said, if you had a good friend truly in states like Oklahoma and Arizona, and if you were interviewing for a position in the plant path department, they would advise you to not take up the position if they ask you to work on this disease. So it was a notorious pathogen to work with. And I went in there and I realized what it was all about. And my advisor there was very, very supportive. He knew that this was a very difficult pathogen to work with, but it was of interest for the local folks. And that's what Noble Foundation was all about. So they had the funds. If I put out a very disruptive idea, they were willing to embrace it. And that's how I explored the options of RNAi. I explored the options of microbiome research. And when I figured out the role of the interactions of those microbes in the soil, not just a standalone pathogen, plant and pathogen interactions, but that holistic environment, I realized that there was so much to learn. And once I finished the projects at Noble Foundation, there was an opportunity that opened up at Trace Genomics, and with my interest in microbiome and their interest in microbiome, we felt it was a perfect match. And it always worked. And because of my interest in always creating a product or doing research that is going to translate into a product that's going to be applied in the field, that was the greatest motivation for me to again move back to industry from Noble Foundation and work on the technology development. I still love doing research. I can't be a complete product or commercial salesperson. So I think I'm in that perfect position where I get to do the research and align it with the market needs.
Exciting. And I want to delve into your interest in microbiome, your research in microbiome in just a moment. I'm curious, though, if in terms of your experience as an industry professional, what do you think is one of the most common misperceptions of industry careers in plant pathology?
When I was in academia, a lot of the research that I did was very fundamental knowledge, which I think is very important for us to understand, answer those questions and move forward towards a practical question. For example, I was working a lot on plant pathogen interactions. I was trying to understand the molecular basis of that process, because I think that's very fundamental for us to understand that. But moving forward into industry is driven by economics. I mean, that at the end of the day makes a difference, right? Everything's driven by economics, but it is highly impact oriented. And for anything to be impact oriented and driven by economics, the solution has to be scalable. So I think that's where the difference lies. A lot of the R&D that I do right now is keeping in the mind about the kind of impact it has on the field, on the customer. Now, in a lab setting, everything is picture perfect. We look for that perfect science, the solutions, the answers to those questions. But when we take the solution into the field, there's so many environmental factors that make a difference. And it's never as picture perfect, although we would love to have that. So when it comes to the field, it has to be impact oriented. So I think it's that frame shift in the mind from perfect science to that impactful science. That is the major difference that goes between academia and industry. And part of that is also because in today's day and time, there are so many challenges, pressing challenges, and we are racing against time. So sometimes even if the impact is 20% or 50% or 60%, I think we should be going for those solutions, and we will evolve and we'll get better at our solutions. But that is the major difference that I perceive in industry versus academia.
That's really interesting. So impact matters here.
Right. Impact is the term, Jim. Yes, impact matters. And is that impact significant enough? It does not have to be the best impact, but if it's significant enough, then it's ready to go out into the market.
Yeah, it's great. So you mentioned it's been 20 years or so since you were in grad school, and no doubt you've grown as a professional. You've learned a lot in that period of time. If you could go back and have a conversation with yourself back as a grad student, knowing that you would end up in a successful career in industry, what advice would you give that person?
The first thing that I would give is have confidence. I think the number one trait that I feel is having confidence in your own capability. Don't have to be perfect. You don't have to know everything, but just have confidence in what you do. And that is going to take you a very long way. I think that's one thing that if I had to have that conversation with myself, I would say that. The second thing I would say, tell myself is clarity on what I want to do is the second thing. I think these are the two big things, clarity and confidence.
Good advice for all of us, actually. I'm curious, you mentioned that Barbara Valent had quite an impact on your career and your professional growth. Are there other mentors that have been really critical in your growth?
Yes, I would definitely name at least three of them. One is, of course, Barbara Valent. I think she had more confidence in me than I did. I think that was the one that really, I wouldn't have been here if not for her mentorship. Forrest Chumley, who is Barbara's husband, but also the Associate Dean of Research at Kansas State University, a very good friend of mine. I think he was another person who always inspired me. I could have a conversation with him at any point of time and gain my own clarity. I think he was a great mentor. Dr. Jan Leach, who was also on my advisory committee, PhD advisory committee, was another person who had a huge influence. I think just looking at her passion to science in the classroom setting, the way she taught, the way she would ask questions, I think those were the things, that inspired me so much. And I will always be grateful for having them on my advisory committee board.
That's great. Mentors are so critical to our growth. And I have to mention, I have the privilege of working with Jan Leach now at Colorado State University, and she actually was a guest on Plantopia earlier this season. So really an individual of immense impact. But
Absolutely. Absolutely. I remember the only image I've ever had of Jan is this beautiful, broad smile, no matter, when I saw her in the hallways. It was always a pleasure to be around with her. I know she moved to Colorado State halfway through my program while I was still at K-State. And today I feel so honored and so privileged and humbled to sit on that Phytobiomes Alliance committee along with her. So she's had a huge role as well in my career.
Well, cheers to all the great mentors out there. Now, I'd like to talk a bit more about science. You've mentioned microbiome, and you've talked about microbes in terms of positive influence on plants. Could you tell me a little bit about what the microbiome is to start?
The microbiome is a very, very broad term. It's all the microbes that live in association in a given environment. So when we talk about phytobiomes, it could be all the microbiomes that are all the microbes that are on the leaves, that are on the stem, that are in the roots, interacting with the soil. So microbiome is a very broad term where it's the biosystem of all the microbes interacting with each other.
So that includes pathogens.
Everything. It includes everything, all kinds of microbes.
So thinking about plant health holistically as you're describing it, what are some of the positive benefits that microbes play in plants?
I think microbes play a huge role in every single process, whether we talk about nutrient uptake by the plants, with all the fertilizers that we're putting in there. Microbes play a critical role, a key role in making them more plant available. Microbes play a role in greenhouse gas emissions. Microbes play a role in maintaining water quality. They play a huge role in plant growth promotion, in yield benefits. So you think about every aspect of they play a very, very critical role in soil texture. They play a critical role in soil health. So the whole any aspect of micro plant and the agricultural systems we talk about, they have a heavy influence. Again, you talk about microbes, they also cause diseases. And I always say, I think there's maybe less than 1 or 2% of all the microbes that are present in our planet that we have ever identified. And out of that, maybe just 5% or less are the ones that cause diseases. All the others are involved in other processes, which we probably don't know the complete landscape. But what that tells me as a person is that the microbes are there not to bother me, but most of the microbes are there to help me. And just as I always keep saying, if I think about it, we are all that topsoil in the making, and microbes will play a role in that in the future.
Yeah, microbes are certainly important environmentally and to plant health overall. Of course, there's a lot in the press these days about the human microbiome and the influence of gut microbes, for example, on all kinds of characteristics in humans. And so it does make sense that same sort of impact is happening in plants as well. I'm reminded that there's a long history of commercial agricultural inoculants. Could you tell us a little bit about inoculants as they've been used historically, some of the successes and challenges of those technologies?
Yeah, historically, there's been they've been there for quite some time, but I think in the recent years, there's a lot more of those coming into the market. Now, the whole space of biologicals and inoculants, biostimulants, relatively, I'm new to this area. It's just been the past few years I've been working on in this area, but it's been a huge learning curve. So the biggest challenge there, the greatest challenge is adoption and trust by the growers. And this is something that I keep hearing over and over and over again. There's one farmer that'll come and say, hey, this microbe worked great. I had this yield benefit. Then someone else tries it and they don't get the benefit. That inconsistency in the performance of this microbial inoculants in the field is the greatest challenge. And I feel like that in itself is the greatest opportunity for us as science to explore. Because if you think about a chemical molecule, no matter where I put it in a desert, I put it in a rainforest, I put it in an agricultural land or an urban atmosphere, it will predominantly perform similarly because it's the chemistry. But a microbe is a living consciousness. I say it always has a mind of its own. It's a life form. So when I take a life form and introduce into an ocean of life forms there, it has to establish itself and then it has to perform. And a life form will always get impacted by every single aspect of the changes in the environment as well. So there is a lot more. We can never treat a biological like a chemistry molecule. So the kind of research, the kind of support system that we need to bring about for these inoculants, the biologicals to be successful is a very different landscape than looking at it from the lens of how we've brought in the chemical inoculants into the market space over the last few decades.
That's really interesting. It brings us to Trace Genomics and your current role. I understand that the approach you're taking is a little bit different than the traditional inoculation. Tell me a bit more about what Trace Genomics does and what your approach is all about.
So Trace Genomics, we bring in the chemistry, the physics, and the biology together because we feel like if we need to look at agricultural systems, we have to have a holistic approach. We cannot look at the microbiome by itself and the chemistry by itself. It is high time we integrate all these parameters together. So at the core of our technology is the metagenomic pipeline that we have. We do whole shotgun metagenomics of the soil microbiome. And we look at, and from that, we integrate that with the chemistry. So we do the chemistry analysis and we do the carbon. We have a carbon measurement capabilities, and we also have a soil texture measurement capability. So we bring in all these different aspects of the soil together. And from that, we've developed what is called as the Trace Environmental Soil System. It's called the TESS. It's the analytical engine. It's 6 to 7 years of our working on this bioinformatic pipelines of the metagenome sequencing and integrating all these other layers. We've come up with this engine, And this engine is the one that propels all the actionable insights that we put out for our customers. And we have three different customers. You can think about it as three different verticals. Our big customer is the ag retail. And for them, it's about, the big ag retail, it's all about yield. For the grower, yield is what that matters. So for them, it's all about disease management, nutrient input, or any other kinds of inputs on the farm. So we combine the chemistry, the carbon, the texture, and the biology data, and we give them a holistic view of what's happening in the field in a manner that they can take actionable insight. Should they put phosphorus liberating agents? Should they put phosphorus biologicals? Where would they expect the performance to be better? What kind of pathogens are in the field? What kind of preventive measures should they be taking? What kind of management and cultural practices would help them? So it's looking at from a very holistic viewpoint. That's what we do for our ag retail. And as part of our technology, what we also do is we give them a seed solution guide based on the crops and diseases, so that it's going to be easy for them to sit with their agronomist or their, whoever they get their seeds from, and figure out what is the best hybrid variety selection, or what kind of crop protection or seed treatments do they have to do. So this is almost, if you can think about it as a one-stop shop, where you get, a very holistic view of the soil environment. The other vertical that we have is we cater to the ESG goals of the CPG companies. As we are entering into the world with, as I always call, the world is in a state of transition. We talk about climate change. We talk about the changing needs. We talk about the soil erosion. We talk about the challenges with our water resources, nutrient, fertilizer, challenges that we have seen in the last couple of years. So, and all the customers also, in fact, I myself, as a customer, when I go to shop, I look for goods that are, made in a more sustainable manner, or fair trade, options is what I look for. So for the CPG companies, they are looking at sustainably produced products, and agricultural products, flood the whole market. What we help is for them to meet their sustainable goals. We look at the sustainability of the soils. So we look at the carbon, and we look at the emissions. We look at the capability of the soils to hold nutrients. We look at the capability of the soils to hold water. We look at the biodiversity of the soils, and we give a sustainability score. So it's a very objective score for these companies to figure out how they're doing. The other third vertical that we have is biological product companies and agricultural input companies. It could be chemical or biological, but given how the market space is right now, we work with a lot of biological and biostimulant companies. And here, the biggest challenge, as I mentioned a few minutes ago, is gaining trust. That is the one word that resounds everywhere when you go and talk to growers, is trust. It's very interesting that a lot of the surveys that have been done in the last few years, the farmers say that we are willing to try, but we don't think we are seeing a response yet. We don't trust that the product is going to work. But they're open to trying. And all they say is, we need more education. We need the data to convince us that this is going to give me a benefit. So I think the onus, I feel like, is on the research community to help that framework. I think we need to understand how to develop that framework, because I don't think we have it. We haven't done enough work to show when a biological will perform or where it'll perform.
Across, environment, a farmer's field, for example, I would expect that over pretty short distances, you could have very different microenvironments that could influence the impact of the microbes. How does your approach deal with that? Is it through more extensive sampling, or how do you tackle that natural variation that we see across the landscape?
Right. That is a very common question we get, Jim, from a lot of our customers as well.
I thought it was a very insightful question. I didn't know it was so common.
Yeah, it is very common question, actually. It is true that there are different kinds of microbes, but when you look at the functional microbiome, when you look at their functions, those differences collapse. We have done several studies. for example, one of the studies, we have gone to Iowa, California, and Texas, and we have done sampling every 4 weeks for 2 years. And then we looked at the microbiome changes. What's very interesting is, yes, there's a seasonal variation. When there's a rainfall event, post-rainfall, there is a change. But the microbes, who is in there varies, but what do they do does not vary as much. Now, when we're talking about an agricultural landscape, if I'm a farmer, I want to know what is the capability of those microbes, to help nutritional support, nutrient cycling. Will they make nitrogen more available? Are there phosphorus solubilizers? Are there phosphorus mineralizers? Are there plant growth promoting microbes? And when we did our extensive studies, what we have seen is that they don't change much. In fact, phosphorus solubilizers and phosphorus mineralizers are one of the most stable populations across geographies. They will shift a little bit based on your cultural practices, based on if there's a major drought. What we've also seen is that the nitrogen microbes in that nitrogen cycle are definitely way more sensitive to environmental perturbations. But what we've also seen is that when temperatures come back to normal, the microbes also come back they normalize as well. So yes, the microbes will differ, but again, in the ocean of microbes, I'm not concerned about what all those microbes are doing. Because first of all, we don't know what all those microbes are doing. The microbes that are very critical for my functions in the field, how do they behave? And it's very interesting to see how the difference collapses once we go to the functional microbiome. And I think therein lies the strength of Trace Genomics as well, is because early on, our co-founders, 2 postdocs from Stanford, were the co-founders of this company. They decided they'd go and use the metagenomics platform instead of the amplicon sequencing. That was a big decision for them to make because amplicon sequencing was less expensive compared to the metagenome sequencing. But then they had this long-term vision of the functional microbiome, because it's the functions that are more important rather than knowing who is in there. And they've invested in this technology. And I think we are really seeing the benefits of that now.
That's really interesting. And you've mentioned the functional microbiome, but you're also equating that to metagenomics. So are you actually testing function, or are you inferring that from DNA sequence?
So just to take a step back, we do whole shotgun metagenome sequencing. So we don't amplify a small piece of the genome, but we amplify the whole genome. We sequence the whole genome. So what that gives us is, it gives us the information of the genes in the microbe. So when we say the functional microbiome, let's say if I have a given soil sample, I look at the gene pool for all the genes involved in the pathway of phosphorus solubilization, phosphorus mineralization, free living nitrogen fixation, nitrification, denitrification. So I'm actually looking at the gene pools in that particular soil and saying, here is the potential of phosphorus solubilization in your soil. Here is the potential for denitrification in your soil. And we say, hey, if your potential for denitrification is high, maybe that's where you should be putting your nitrification inhibitors. That's how you place your product, a smart way to place your product. If your microbiome for phosphorus solubilization and mineralization is doing great, you have really good microbiome there, keep doing what you're doing. You're doing the right thing there. So it's all about looking at the functions of that microbiome by looking into the gene pools of that particular soil.
And so ultimately, you don't care who's fixing nitrogen, for example. You're looking at the broad consortium that's present and asking, does this consortium have that capacity?
Yes.
And then you're making grower recommendations based on those observations.
Right. So here is another caveat, and I always mention this. I always say nitrogen fixation potential or denitrification potential, because we're measuring the genes, we're not measuring the transcriptome. Is it actually doing it or not is not what we're measuring. But what we have seen repeatedly over and over in our data sets is that when we get soil samples from farms where they've done very progressive and regenerative practices versus a very conventional monocropping heavy tillage systems, we very clearly see the difference on the gene pools. So we have validated our technology, with our customers and on-field data sets. But yes, what we look at is the gene pools, and yes, we don't ask who is performing the function. The question is, are they performing the function?
So the technology, the broad holistic approach that you're taking reminds me of IPM in some ways. Obviously, the goal is different, but really you're synthesizing information from multiple different disciplines to come up with a single unified approach that helps us leverage microbes for the benefit of plants.
Yeah, that's a good way to put it. And there's another way also I put it is, when you think about the microbiome and the technology that we do, I say we are the 23andMe for soils.
Oh, I like that. Might run into some trademark issues there. I don't know. That's certainly something I can relate to.
Yeah. So
it sounds as though the field of phytobiome is progressed a lot in the last 10, 15, 20 years. When you're looking forward and thinking broadly, not only about what Trace Genomics is doing, but the broader agribusiness industries, what do you envision as some of the biggest opportunities that are still out there for agriculture?
I think one of the real unexplored area for us in the field of agriculture, I think, is that holistic soil health. Is something that we haven't paid much attention to. I think there's been a lot of emphasis over the years in terms of inputs for us to get out a certain output. And over the years, what we have seen is that soil health had degraded. And part of the reason I think why that has happened is, in spite of our best efforts, is that we have never paid that attention to the soil life. We've looked at soil as chemistry. And more recently, the Haney test had come through in the recent years, but Haney test was a good move towards the biology, but it is still not comprehensive. I think where we are heading into is, if we really need to preserve our agricultural landscape and improve that landscape to meet the challenges and the needs of our world in the future, we really need to restore our soil health. And I think the way we have been doing our soil diagnostics has to change. I mean, think about it. Technology has changed. Agricultural practices have changed. But for the past 70, 80 years, the only traditional test we've been doing is soil chemistry. I think it is really high time that we move and integrate a lot of these technological advancements, which have been in the academic area, to come into the field. And I think moving into the future, I think there's going to be a huge revolution from where I'm standing, where I see is a huge revolution in the field of agriculture driven by data analytics.
I'm excited about that. Can't wait. So, Prasanna, thanks so much for being our guest today on Plantopia and for talking about industry careers and soil health and the phytobiome.
Thank you, Jim. I really enjoyed the conversation.
I did too. Thanks so much. You just heard from Dr. Prasanna Kankanala, Director of Research and Development at Trace Genomics. And I'm Jim Bradeen, the host of Plantopia. Thanks for listening.